Now we're going to take a close look at the reactions of the sea urchin egg surface to fertilization, to the arrival and fusion of the fertilizing sperm, and how the egg prevents more than one sperm from entering. Now I'm going to draw across the whole board a time course of transformations that the egg membrane undergoes as the sperm binds. Now, the egg comes with a pre-made envelope right on top of the surface, which we call the Vitellin envelope. Doesn't matter what it means, that's just what we call it. Whereas this bottom layer here, that's the egg cell's plasma membrane. Now, the sperm binds and actually fuses with the egg cell membrane at some point. There's the acrosomal process of the sperm leading up to a head. Recall that during the acrosome reaction, the acrosomal vesicle fuses with the sperm plasma membrane in response to a rise in calcium. This is just an instance of the cell's regulated secretory pathway and is fundamentally similar to the process that goes on in a synapse and one of the cells that secretes insulin, etc. And a very similar reaction takes place in the egg as it's fertilized. The egg contains, docked right underneath the plasma membrane, an array of large packed secretory granules called, spectacularly imaginative phrase, cortical granules, because they're granules at the cortex. These are secretory granules packed with something and they are just waiting for a calcium rise to fuse with the plasma membrane and dump their contents. Now, this diagram is going to go across about 5 minutes of time from the time the sperm binds. So on the scale of the board, 1 minute is roughly about that long, okay? Now, not long after the sperm binds, there are a number of electrophysiological events, but within 15 or 20 seconds, there is a calcium rise in the egg cell cytoplasm, starting at the site of sperm entry. So calcium goes up abruptly at about this time, and that triggers fusion of these cortical granules with the egg cell membrane, thus liberating their contents to the outside world. Their contents include a bunch of enzymes that snip away the bonds that hold this Vitellin envelope to the egg cell surface. They also include proteases that chew away the receptors for sperm. So once these things have had their way with the egg cell surface, sperm won't be able to bind to it anymore. And all this membrane added to the egg cell surface, what's going to happen to that? That's a whole lot of surface area. It looks very wrinkly and so on for a while. And soon something else begins to happen. one immediate consequence of the cortical granule reaction, of the contents that they put into the extracellular space, is that this Vitelline envelope is released from the egg cell surface, and under the influence of osmotic pressure begins to elevate above the egg cell surface to form a kind of a balloon around the newly fertilized egg. At this point, it ceases to be called the Vitelline envelope, and we call it the fertilization envelope or fertilization membrane. It is the visible site, again, for those taking the lab, it is the visible site that an egg has been fertilized, and its elevation starts right around the site of the fertilizing sperm. Now, again, I mentioned their contents. The cortical granules also include a protease that chews away sperm receptors, and it includes peroxidases that harden this envelope into a cross-linked protein mesh through which large molecules can't get through. Water can penetrate, but not proteins, for example. So once this is established, no more sperm are going to bind. The final thing in the cortical granules is the material for a new extracellular matrix. But before we get to that, recall this is the process of exocytosis, just like what was happening during the acrosome reaction in the sperm. That adds a lot of membrane surface area to the egg cell, and it is rapidly followed by endocytosis. That's what you call it when the cell gulps up a bit of membrane into a vesicle like this. So endocytosis, within a few minutes, takes up the extra membrane released by the cortical reaction. Some of this material released by the cortical granules begins to form a kind of a gel right over the egg membrane. The egg membrane starts to grow a dense forest of microvilli, and you guessed it, those are based on the extension of actin filaments, as is, in part, this process of reuptake endocytosis. These endocytic vesicles acquire quickly an actin-rich coat, which contributes to contracting them down into little nubbins eventually. They join what's called the endosomal membrane system and are recycled eventually. These objects are endosomes. This material, this new extracellular matrix, is made of a protein called hyaline, and the gel that they compose is the hyaline layer. In sea urchins, brittle stars, and some other echinoderms, this hyaline layer acts as a kind of a shellac that holds the cells of the early embryo together until the time when they can establish real junctions between them. This is the visible part of the process. And this is known as the slow block to polyspermy. And it's permanent. There's no reversing this. But it takes about a minute to get going, during which time many other sperm might have bound, and therefore eggs also have a fast block to polyspermy. that is active during this first minute. And the fast block starts within a few milliseconds of sperm fusion and is based on membrane electricity. In short, sperm fusion triggers a rapid membrane depolarization. It reverses the voltage across the membrane, but the cell can only sustain that for so long. Now, let's move to the other board to talk about what that means. To understand the fast block, we need to talk about some of the electrical properties of the cell membrane. The first point, lipid bilayers, and I'm going to draw a sequence of lipid bilayers on the board here, top to bottom. Lipid bilayers are intrinsically permeable to things that are nonpolar. CO2, O2, gases like that, or lipids, or whatever, oily things, steroid hormones. They come and go freely across the cell membrane. The membrane is highly permeable to these things. Water, of course, is polar, but water is small. The cell membrane is not impermeable to water, nor does it conduct water across very quickly, except through specific channels. So the permeability of small, albeit polar molecules like water is low but significant, whereas the membrane is essentially impermeable to ions, also to large polar molecules such as glucose. Want to get glucose into cells, it has to be transported across the membrane somehow. ATP, well, it'd be great if you could get ATP into cells, but actually the point is you want to keep ATP inside the cells. Likewise, glucose, once you get it. So there might be a lot of these things around, but the cell membrane is basically impermeable to them, and that goes for stuff that the cell manages to hoard inside itself too. After all, the whole point of a cell is to concentrate good stuff that it wants to use on the inside and not let it just leak away, right? Okay, so if cells are full of solutes, good stuff like ATP and glucose and proteins and DNA and whatever, and water can just seep in, sooner or later it's going to blow up just due to the osmotic effect of water dissolving the contents inside, okay? So the fundamental solution possibly the single most important protein in all of our lives, is this remarkable pump that burns ATP in order to exchange sodium and potassium ions. I'm going to draw potassium in green. Seems kind of like a greenish ion. And sodium in blue. Sodium ion binding site on the cytoplasmic face. And this protein hydrolyzes ATP. That is, it uses the chemical energy of ATP hydrolysis to pump potassium ions in to the cytoplasm while exchanging them for sodium ions, which get pumped out. Okay, so potassium goes in, and it happens that this pump takes 2 potassiums across for every 3 sodiums. Consequently, This is the outside of the cell, and this is the inside of the cell. Sodium is high outside, living cells. Potassium is low, unless, of course, you happen to be in some sort of funny brine pool. Sodium in cytoplasm is low. Potassium is high in the cytoplasm, typically about 20-fold higher than it is on the outside, and roughly the opposite for sodium. Your cells, depending on what kind of cell they are, spend 30 to 60% of their energy budget running this one protein. From a certain point of view, more than any other single thing, that's the protein that makes a cell alive. And it maintains this sodium-potassium chemical gradient. So these are the sodiums again, whereas these are the potassium ions. So since the dawn of life, cells have relied on gradients like these, like this, to do useful work. You've probably all heard at some point in a previous biology class, for example, that the mitochondria use a proton gradient across the inner membrane to drive a spinning rotor that synthesizes all of this ATP. Well, most of that ATP is in turn getting used to drive the sodium-potassium ATPase. There's a simpler example that's perhaps a little easier to explain than the mitochondrial electron transport and proton pumping mechanism. I'm going to diagram a transporter protein, sort of a pastiche of what a transporter protein might be like, really. Such proteins really exist. However, for example, in your intestinal cells, where the epithelium of the intestinal wall might want to take up, say, dissolved sugars, such a protein might have a sugar binding site facing the outside world, and also a sodium binding site. And let's say it's rigged up so there's sugars high out here and, digested from food or whatever. And what color were my sodiums again? I think they're blue. Yeah, they're blue. Sodium is blue. This is outside a cell, so there's lots of sodium kicking around out here. Now, let's keep in mind there's lots of sodium out here, but depending on what you ate for breakfast, glucose isn't necessarily all that high out here. In fact, the whole goal here is that glucose get concentrated inside your cells so you can make use of it. You're taking up glucose typically against its concentration gradient. That is, there's more glucose where you're trying to put it than there is where you're trying to get it from. And of course, there aren't so many sodiums around, right? So imagine that this pump is rigged up so that, yes, it binds a sugar molecule, but when it binds a sugar molecule, enough of them, now I know you've seen, some of you at least have seen a diagram like this before. The idea is that if it binds the right combination of things, it binds sodium and sugar molecules together, it'll change conformation dumping sugars into the cytoplasm at the cost of transporting a few sodium ions in there as well. In this case, the sodium is moving down its concentration gradient because, again, sodium is high out here and low inside. So sodium in this case is moving with its concentration gradient. That reaction is favored, and it is being harnessed to do the useful work of taking glucose against its concentration gradient. This is a cartoon protein, but essentially this effect is the basis for the design of Gatorade. Next membrane. In addition to transporters, the membrane is full of channels that let ions through or not. So we've got pumps, transporters, and channels. The key channels of interest here are ion selective channels, especially ones that are gated by voltage or by ligands. Of course, that's an easy icon for a closed channel. Here we have an open channel. Which might allow, for example, let's consider sodium again. It might allow sodium ions specifically to flow from outside the cell into the inside, which they will do because this happens to be both down their concentration gradient and, as it happens, down their electrical gradient. Many ion channels are gated by the binding of ligands. This is how many neurotransmitters work. The channel might be blocked, for example, by some sort of plug that is relieved by the binding of some kind of ligand. Whatever it is, some sort of neurotransmitter, let's say. Again, this allowed just to be different. We'll have it be the potassium ions that are flowing out this time. So ion channels, many of which are gated by ligands or, for example, by membrane voltage, are key to regulating the permeability of the membrane to ions. Now, ions are charged. Separation of charges creates an electrical potential, that is, a field that impels charges to move. If we just imagine the sodium ions themselves, the extent to which there's more positive charges out here than there are over here is results in a force impelling those charges to move that way, or some other like charge that will equalize charge distribution on both sides of this barrier. That is, the membrane can act as a battery or a capacitor, storing power as a charge difference across an insulator, as long as the membrane remains impermeable. And in animal cells, the resting membrane potential results from basically the conjoint operation of the sodium-potassium ATPase combined with a potassium leak to the outside world. And that's going to be the last little iconic membrane that I draw here. So here's the sodium-potassium ATPase. Combine that with a channel that lets a little potassium leak out. So potassium going in, again, 2 potassiums going in for every 3 sodiums going out. This exchange continually maintains a chemical gradient for both of these ions. That is, there's a lot of sodium out here that wants to get in, and there's a lot of potassium in here that would tend to get out if it could, some of which is leaking through this leak channel down its chemical gradient. But as it does so, it's leaving behind a slight excess of negative charge in the cytoplasm, which leads to a membrane potential, which in a resting cell is something like, well, it varies, but we'll just use the ballpark value of about 70 millivolts. So the sodium-potassium ATPase establishes strong chemiosmotic gradients of both ions, with sodium high outside the cell and potassium high on the inside. Opening a potassium channel allows potassium to flow down its chemical gradient, down its concentration gradient, which means out. That leaves behind unbalanced negative charge, which creates an electric field that opposes further flow of positive ions, the potassium ions, out of the cell. So in a resting cell, this equilibrates somewhere, and let's say it's something like 70 millivolts, which is about appropriate for a nerve cell, or as it happens, for the egg. Now, we'll turn back to the egg. Before fusion, here are the eager suitors. Sodium is high out here. Potassium is high in here. And the membrane potential is strongly negative. Channels are closed, the resting potential is stable. Step 2, one sperm fuses and a little sodium goes in. Where's my blue? There's blue. A little sodium gets in. The potential rises a little bit, perhaps to negative 50 millivolts. Voltage-gated sodium channels in the plasma membrane of the egg open in response to this slight change in the membrane potential and voltage-gated sodium channels will let a lot more sodium in. And by a lot, I don't mean a lot relative to its concentration gradient. I mean a lot relative to the electrical potential. It turns out to change the electrical potential requires a tiny fraction of the actual concentration gradient of sodium or potassium. So voltage-gated sodium channels open, the membrane potential rises to about positive 20 millivolts. For some reason, this membrane fusion process works at electrical potentials that are below about negative 20 millivolts, but not above. How? I have no idea. I'm not even sure anybody has any idea. But it's not hard to imagine how you could design a membrane protein that had parts that were moved by an electrical field and therefore would be sensitive to the electrical charge across the membrane. After all, we know lots of them, the voltage-sensitive ion channels that are the basis for axon conduction and for things like this. Okay, so surely it's possible for us to imagine that the fusion proteins in the sperm and egg could also be voltage sensitive. In fact, many proteins might be, at least to some extent, voltage sensitive. Now, once the potential gets positive, these voltage activated sodium channels have the feature that they inactivate and won't be openable again until the membrane returns to its resting negative potential. So step 4, the sodium channels inactivate. Here we've got our fertilization cone and the sperm is in there now. There's its tail sticking out. And I guess I forgot to draw the losers in here, looking sad. And they know that's what's coming for them next. But the potassium channels open in response to this positive membrane potential and let potassium out. Whoops, wrong color. Letting potassium out, again, ultimately drops the membrane potential back down to 70 millivolts as the sperm is fully engulfed, the fertilization envelope starts to rise, and here's the sperm head in there again. And what happens next, a little bit slower, is that calcium ion concentration in the cytoplasm goes up. As it is released from intracellular stores. Perhaps a little bit gets in at first due to voltage-activated calcium channels. Perhaps there's another mechanism, something like that. At any rate, the calcium rise now takes us where we started, back to the slow block, to polyspermy and the triggering of exocytosis of the cortical granules. Now, I want to make one little correction here. I said in the egg, at a positive membrane potential, potassium channels open and they let potassium flood out of the cell to restore the membrane voltage. Now, that's what would happen in a nerve during axon conduction, okay? It's not actually clear to me that that's what happens in an egg. It may just be that the leak of potassium slowly reestablishes the resting potential once the sodium channels have done their bit. What's happening in the egg is an action potential. It involves a membrane depolarization due to the abrupt threshold-dependent opening of sodium channels. But it takes place on a much longer time scale than does axon conduction in a nerve. Okay, so the conduction of nerve impulses takes place on, in comparison, millisecond timescales. And to finish this particular topic, I'll just draw, for clarification, a cartoon of what happens once a nerve has already committed to firing an action potential. Of course, one big difference here is that the axon is a linear structure. It might be very big in the case of something like the squid giant axon, where much of this physiology was first characterized, or it might be very small, like in many of our nerve axons. At a particular patch along the axon, as the action potential is conducted, we can just divide this into chunks. I'm going to just limit myself to 3 of them here and say, this middle one is where the action potential is currently going on. Sodium is rapidly flowing in, depolarizing the membrane. And it's spilling out little blue sodium ions, spilling in everywhere, right? They were supposed to be blue, weren't they? Yes. Okay, so sodium is going in, depolarizing the membrane. That means in the part over here where the action potential just passed, potassium is going out. And the sodium channels have inactivated. Whoops, excuse me. Right? So sodium in. Recovery from the action potential means potassium is flooding out until they reach the resting potential again. All these ions are entering the cell, diffusing around, and in this patch here, it's the slight voltage change from the entering sodium that triggers the opening of sodium channels, voltage-dependent sodium channels in the next patch, and thus propagates the action potential down the axon. Well, once it gets to the nerve terminal, guess what happens down here? Well, if I magnify this a little bit, it looks strikingly similar to what I drew in the egg. In the synapse, secretory vesicles are just waiting, docked at the plasma membrane, just waiting to dump their contents into the extracellular space. When calcium rise comes along, in the vicinity of the synapse, all those secretory vesicles do just what they're primed to do. Not all of them, only a few actually fuse at once with the plasma membrane for each action potential that comes to the synapse. At any rate, some of them fuse with the plasma membrane, dump their neurotransmitter across the gap, trigger some reaction perhaps in the cell on the opposite side, and then immediately exocytosis takes up the membrane again, and these vesicles are in fact rapidly and locally recharged with neurotransmitter to prepare to release again. So very much like this picture, the arrival of an action potential at the nerve synapse triggers, in this case, triggers voltage-dependent calcium channels to open, and they actually let calcium in from outside the cell to trigger the exocytosis of docked secretory vesicles. That brings up another major difference between these two events, fertilization's action potential versus the action potential in a neuron, and that has to do with the source of calcium. As I mentioned, calcium is probably flowing through a voltage-gated calcium channel at the synapse, when the action potential arrives. However, in the egg, much of the calcium that rises during the calcium wave that follows fertilization and stimulates cortical granular exocytosis is probably coming from intracellular stores, endomembrane tubules, part of the endoplasmic reticulum, are wrapped around the cortical granules as they are through almost everything else in the cytoplasm. The endoplasmic reticulum actively sequesters calcium and releases it rapidly into the cytoplasm in response to a couple of different things, notably a small molecule called inositol trisphosphate, which is a phosphorylated sugar cleaved off of a membrane lipid in response to various signaling events. Why calcium? So here's why I think why calcium. At the dawn of life, all the other ions were busy. Cells figured out how to pump sodium and potassium back and forth. You need magnesium to hydrolyze every ATP. Magnesium is a participant in so many reactions inside the cell. Well, zinc and iron, they're precious. They're useful for enzymes. There isn't that much of them around anyway. Things like sulfate, carbonate, they're great, but they're not terribly useful for this kind of thing. Calcium is just the ion that wasn't busy, so it got to send all the messages.